Welcome to the presentation. My name is Fisa Irfan and I am student of Master of Medical Biotechnology. I am presenting the analysis of stress in chicken through measurement of glucocorticoids in serum, feather and eggs. What is stress and stress response? There is an increasing amount of interest in the welfare of chicken and the importance of stress and stress responses in chicken welfare. Stress is a complex process that has been defined as a disruption in homeostasis by stressors which trigger numerous defense mechanisms. Stressors are the harmful stimuli which provoke a series of events known as stress responses. There are two kinds of stress responses, physiological response and behavioral response. The behavior response involves active responses and passive responses, which involve fight and flight and abnormal behavior of an organism, while the physiological response involves the activation of two systems, sympathetic adrenal system and HPA axis. The sympathetic adrenal system involves the release of adrenaline and non-adrenaline, while HPA axis involves the release of numerous hormones. Glucocorticoids is one of these hormones. Stress responses are not always detrimental, but are normal occurrences which are essential to the survival of an organism. However, when exposed to a longer period or repeated incidences of stress, it can affect the biological functioning of an organism. The aim of this study is to discuss the physiology of glucocorticoids, to analyze the pros and cons of methods for measuring stress hormones in chicken and different sampling requirements for chicken serum, feather, and egg, and analyzing the gap in research for the exact measurement of stress hormones. What is glucocorticoids? Glucocorticoids are small hydrophobic hormones which are not stored in cells but are produced from cholesterol upon stress. They are then released in blood bound to a protein such as corticosteroid binding globulin or albumin, where they are inactive and can only be active by dissociation from their carrier protein. After migration to target cell, they can exert action after binding to type 1 and type 2 glucocorticoid receptors. The mechanism of glucocorticoid production involves the release of glucocorticoid by the activation of HPA axis. Initially, cells that produce the neurotransmitters are activated by stressors, causing the release of corticotropic hormone from hypothalamus. This in turn activated the pituitary gland, which releases the adrenocorticotropic hormone, and in turn and in the end it activated the adrenal gland. The adrenal gland will then activate it to release cortisol or corticosterone into the blood. The adrenal cortex is comprised of three layers of epithelial cell, each layer producing a different hormone. The middle layer is responsible for the production of glucocorticoids. There are two assays for measuring stress hormones, immunoassays and mass spectrometry. There are several types of immunoassays: radioactive immunoassay, chemiluminescence, and enzyme immunoassay. All of them are based on the same principle, in which the analyte of interest compete for the antibody binding site with a labeled form of the analyte that is tagged with either a radioactive isotope, luminophore, or enzyme. While the mass spectrometry, there are two types of mass spectrometry: less liquid chromatography mass spectrometry or gas chromatography mass spectrometry. They are based on the same principle, in which the separation of analyte in a mixture by chromatography and detection by mass spectrometry. Now I will discuss about the differences between these two assays. The mass spectrometry, the LC-MS has two types, low resolution and high resolution. And immunoassays are also two types: indirect immunoassay and direct immunoassay. All of these techniques require sample pre-treatment before analysis to remove all the cross-reacting substances which can interfere the analysis of steroid hormones, while direct immunoassay requires no purification. Indirect immunoassays require solvent extraction before analysis, and while mass spectrometry analysis require extensive sample purification and extraction process. It involves solid phase extraction, liquid-liquid extraction, and solid-liquid extractions. There are certain pros and cons for these two assays, with immunoassays suffering from low specificity and sensitivity because of the considerable amount of cross-reactant substances which can confound the result for the analysis of steroid hormone. Moreover, they can only analyze one steroid at a time, and however, it costs low. On the other hand, mass spectrometry are fast, highly sensitive and specific, and are capable to measure multiple steroid and steroid metabolites. However, it costs more than immunoassay and require expertise. Liquid chromatography tandem mass spectrometry is considered as a gold standard in measuring glucocorticoids in chicken. Tandem mass spectrometry has improved specificity by measuring the intact molecular ion and diagnostic fragment ion produced by the dissociation of intact ion. It shows limitation in analyzing stereoisomer which produce identical fragmentation pattern. Ion mobility spectrometry has shown promising results in separating stereoisomers based on stereochemical and structural differences as an orthogonal separation technique. There are two types of method for measuring glucocorticoids: invasive method and non-invasive method. Invasive method, the process of taking blood sample is an invasive method which confounds the detection of adrenal cortical cell responses because of the additional stress involved in sampling. Blood is a complex matrix containing numerous biomolecules which need to be removed before analysis to improve the specificity and sensitivity of analysis. The non-invasive method involves feather, eggs, and feces. In feather analysis, it provides the concentration of glucocorticoids at the time of feather growth and can provide confounding result because of the local production of glucocorticoids in feather. Egg contain two types of sample, yolk and albumin. And the analysis of egg glucocorticoids is based on the fact that maternal glucocorticoids are transferred to developing embryo, and maternal adrenal cortical activity can be determined by analyzing these glucocorticoids. There are two types of samples which are differ in the time required during the egg development process. While analyzing the blood glucocorticoids, sample extraction and purification process is very important to remove all the biomolecules which are present in blood which can confound the result. The initial sampling requirement for the serum and plasma are different, with plasma requiring anticoagulant and contains fibrinogen, and serum sample don't require any anticoagulant because fibrinogen is absent in serum, which is responsible for blood clotting. And 80% of the glucocorticoids in blood is bound to protein, and 10% of the glucocorticoids are present as free, which is known as biologically active. Feather TCs, it involves the extraction process for the feather glucocorticoids is more laborious than plasma extraction because the sample require mincing and grinding. Small sample, small sample mass of feathers measured high glucocorticoid concentration, while large sample mass measuring low concentration, known as small sample artifact. The artificial effect of sample mass on steroid concentration, showing that samples of different masses are not directly comparable due to methodological defects. And it can also provide the glucocorticoid concentration only at the time of feather growth. Egg glucocorticoids. It was found that the corticosterone are present in very small concentration in egg albumin and yolk, while other hormones are present in more considerable amount than corticosterone, like cortisol. This raises the concern about the biological relevance of egg corticosterone to understand adrenal cortical stress response of mother. Research also explains the presence of metabolic buffer. Metabolic buffer is the mechanism of maternal steroid metabolism in developing embryo, either by completely circumvent the effect of maternal steroid or regulate the exposure of GCs via selective reactivation. Dominant glucocorticoids are age and organ specific. It was found that high levels of cortisol is present in avian immune system of developing embryos as compared to glucocorticoids. Cortisol level decreases with age in avian immune system. Similarly, in plasma, cortisol concentration is higher and decreases with age, and inverse happen with corticosterone. Cortisol should be considered when measuring glucocorticoids in avian, considerable levels are found in chicken egg albumin. Cortisol is also analyzed in feathers and found to be related to avian death. It is concluded that assessing the welfare of chicken by measuring corticosterone concentration in serum feather and egg of chicken has certain limitation even after validation. Chicken welfare should not rely solely on measuring corticosterone. Health, behavior, and ecological measures should be considered for evaluating chicken welfare. Thanks for listening.